Hi, everybody. My name is Ed Davis. I'm Senior Application Scientist at GeneCopoeia Incorporated in Rockville, Maryland, located just outside of Washington, D.C. I'd like to thank you all for joining me for today's webinar entitled Safe Harbor Transgenesis in Human and Mouse Genome Editing. We really appreciate it. Before I get into the webinar, I just want to go over a few practical matters. First, you may ask me questions at any time. However, in order to do so, you will not be able to speak vocally to me because your microphones will be muted. Instead, you'll need to type your questions in the questions box located inside your control panel. If you can't see your questions box, it might be hidden, in which case look for a small strip of buttons on one side of the screen. One of those buttons will be orange with a white arrow in it, and if you click on that button, you'll then be able to see your control panel and your questions box. And secondly, in the interest of keeping the talk going smoothly, I will wait until the end of the talk to answer the questions in the order in which they were received. And when I do so, I will read both the question and the answer out loud so that everybody can hear. All right, so with that, I'm going to go ahead and start the webinar. So before I get going, I just want to tell you who we are at GeneCopoeia and what we're about. We were founded back in the late 1990s as a provider of large-scale human and mouse ORF clones, or open reading frame clones. And these are ready-to-express plasmid clones that are designed mainly for mammalian systems, but also we have them for bacteria and yeast, as well as others. And these are ready to express ORFs with the 3' and 5' ends removed. And literally ready to express, you get them, you can put them into your cells the day you get them. And you can order them for virtually any human and mouse open reading frame. Since then, we have expanded these clone collections out to include promoters, that is human and mouse promoters. These are regions upstream of the transcription start site that are linked to reporter genes like luciferases. MicroRNA clones, those come in various configurations like microRNA 3' UTR target clones for studying interactions of microRNAs with genes. And again, those are reporter clones. We also have microRNA precursors and inhibitors. We have a large selection of CRISPR clones. These are mainly targeted toward each gene, again in human, mouse, and rat. And you can order them right on our website, or you can order them for specific purposes like mutagenesis. shRNA clones if you are doing more traditional knockdown type of experiments. In addition, we have a number of solutions, large-scale solutions for viral systems, mainly for lentivirus and adeno-associated virus, or AAV. In both cases, you can purchase expression clones in lentiviral or AAV backbones, and these can be packaged into virions, or you can have us package them. We have large-scale packaging services. Further, we have a large number of kits and reagents, such as for transfection. We have our own transfection reagent line, luciferase kits for secreted systems like Ghostia luciferase, or the more traditional Firefly and Renoa luciferases. FISH probes are fluorescence in situ hybridization probes, usually for either chromosome counting or enumeration, or for disease gene-specific FISH probes. We have indel detection kits. Those go along with CRISPR genome editing applications for validating sgRNAs. And a number of cloning kits, like recombination cloning kits. And finally, we have a large product line devoted to fluorescent detection. These comprise either cell function assays, such as for apoptosis or autophagy. We have nucleic acid detection reagents, such as DNA gel stains or DNA quantitation reagents. Cell structure probes, so detection reagents for organelles like mitochondria or lysosomes. And finally, we have a large number of fluorescent dyes, which can be conjugated to things like antibodies. But of course, you're here to learn about CRISPR and genome editing related reagents, so let's talk about that. All right, so the main topic of today's webinar is safe harbor. So what is safe harbor in terms of genome editing? Well, these are genome sites that permit transgene insertion with no known adverse consequences on cell fitness or viability. And they also are known to contain an open chromatin structure, so this allows for consistent, stable transgene expression. Now, before I get into more of the background, this is all centered around GeneCopoeia's Safe Harbor products. So these come in two main categories. We have Safe Harbor Knockin Kits. These are kits with validated CRISPR or TALEN, that's an older genome editing technology-based plasmid clones, Knockin verification PCR primers, and Knockin controlled donor clones. They can also come with or without either Knockin donor cloning vectors or pre-made clones for knocking in genes of interest, such as the CRISPR-Cas9 nuclease, as well as the verification primer pairs. In addition, we have what are known as Safe Harbor Knockin ORF clones. These are similar in some ways to the ready-to-express ORF clones that I mentioned before, but instead of being episomal like our classic ORF clones, these are for knocking in genes to Safe Harbor. So these can come with any of more than 35 to 40,000 human and mouse specific sequence verified ORFs in these knock-in donor clones, and they go along with our knock-in kits. All right, so here's a general outline of today's seminar. First, I'm going to talk about transgenesis in terms of applications, and then I'm going to talk about important considerations for transgenesis. Then I'm going to give a brief introduction to Safe Harbor. Then I'm going to talk a little bit about CRISPR and how that works. And finally, I'm going to go into more detail about GeneCopoeia's Safe Harbor solutions. All right, so let's talk about applications. So there are a number of useful applications for transgene insertion. These are some of the general ones. One of them is cross-species expression. So for example, you might want to express a human gene in a mouse, or vice versa. You could express a mouse gene in a human cell line, for example. Another thing you can do is rescue a mutant phenotype. So let's say you did a mutagenesis on a gene in particular and it caused a phenotype. Well, you could knock in a safe harbor or, for example, to rescue that mutant phenotype. So one of the classic approaches in genetics is once you get a mutant, you can put the wild type back in and see if it rescues the phenotype that you see. Another application for transgenesis is gene overexpression. So let's say you want to get large quantities of a particular protein. You could put in a transgene to overexpress that gene. And another one is gene tagging. Let's say you want to add a GFP to the 3' end of a gene. You can do that too. So these are just a few examples of that can be done with transgenesis. And another example, prominent example, I'm going to go into a little more detail is CRISPR library screening. So I haven't talked about CRISPR much yet, but one of the things that people are doing with CRISPR nowadays is to screen CRISPR libraries for either gene knockouts or overexpression. And there are a number of ways of doing this, but Safe Harbor transgenesis is important can be important for CRISPR library screening because these libraries are often use lentiviral clones for expressing the CRISPR guide RNA, and as you may know, CRISPR also requires the presence of Cas9 nuclease, and sometimes people put both the Cas9 nuclease and the single guide RNA on the same plasmid, but when you're doing lentiviral transduction, the titers tend to be lower when both Cas9 and the guide RNA are on the same plasmid, as shown here. This compares the relative functional titer between a so-called all-in-one plasmid where both Cas9 and guide RNA are on the same plasmid versus just guide RNA alone. the titers that are about tenfold higher when you remove Cas9, and that's because Cas9 is large, so it tends to reduce the viral titer. And so one thing that transgenesis can be used for, safe harbor transgenesis can be used for, is to establish a stable cell line using a construct like this, where you can knock it into a safe harbor site, and you'll have a Cas9 expressing stable cell line, which can then be very conveniently used for library screening. And GeneCopoeia has a bunch of pre-made cell lines that stably express Cas9. We have more than 40 now, and we're making additional ones. Or you can purchase a clone like this with our safe harbor system and knock in Cas9 to safe harbor. And that's for human or mouse. All right, so let's talk about some of the considerations for transgenesis. So one of the most important, probably the most, maybe even the most important consideration for transgenesis is that when you knock a transgene into a cell line or into an animal, you don't want it to disrupt a gene that's important for cell growth or other function. Now, you can, obviously, if you get clones that cell line clones or animals that survive insertion, random insertion, they haven't disrupted an essential gene, but they could disrupt some other gene, and that disruption could confound the interpretation of the results. Also, you don't want the transgene insertion to cause tumorigenesis, either by disrupting a tumor suppressor gene or activating an oncogene. Further, insertion should allow genes to be expressed in all cell types, and these insertions should be stable. You don't want them to be lost by either silencing, which can happen with lentiviral transgenesis or some other forms of transgenesis, or you don't want them to be lost through recombination. And ideally, insertion should allow creation of isogenic lines. So in other words, let's say you are knocking in several genes to the same cell line. You want to make different cell line clones, one with gene A, one with gene B, one with gene C. Well, if you insert these transgenes by random integration, they're not really going to be isogenic because the insertion sites are going to be at different sites. So safe harbor allows you to insert a gene at one particular site, and so it can be truly isogenic. So some of the traditional transgenesis approaches don't have all those characteristics. One of the most commonly used approaches is viral integration, usually lentivirus. This is very efficient. It's actually relatively easy to introduce a transgene into cells via lentivirus. And in fact, lentivirus is good because it infects nearly all cell types, which is an advantage over transfection. However, as I mentioned before, integration is random and it tends to favor transcription units. So you can disrupt genes that are important for growth. You can also do random integration using non-viral methods. This is a little bit less efficient than lentiviral, especially in hard-to-transfect cells. Or you can do transient plasmid transfection. This is efficient, but it's usually not stable because the transgene does not get stably integrated. Okay, so safe harbor gets around all of these problems. So again, what is safe harbor? Again, these are genome sites, particular defined genome sites that permit transgene insertion with no known adverse consequences on cell fitness or viability, and they contain an open chromatin structure. All right, so the first widely used safe harbor that was described is from mouse. It's known as ROSA26, and this group found that one strain of mice, known as Rosa beta Go26, expressed beta-galactosidase from a randomly inserted transgene at high levels uniformly in nearly all tissues examined. So they mapped this site to chromosome 6, and they found that the locus expresses one coding transcript and two non-coding transcripts, and only the non-coding transcripts are disrupted by the insertion. Now, I'm sure you noticed this took place a long time ago, more than 20, about 20 years ago. And so back then, people didn't think that there was much of a role for non-coding transcripts. Now, there might be. In fact, there are important cellular biology roles for non-coding transcripts. But still, it was still good to know that at least a coding transcript was not disrupted. Now, they did notice that slightly fewer mouse pups are born from homozygous mothers, that is, mothers with um, the Rosa 26, both copies of Rosa 26 modified, than from heterozygotes, but these pups develop normally and are fertile. So there don't seem to be any problems. And there are definitely not any kind of issues in heterozygotes. So over the years, this has become a standard locus for transgene insertion in mouse, both in vitro, in cell culture, and in vivo. Many years later, a similar type of site was identified in humans. That's also known as safe harbor, and this is known as the PPP1R12C gene, which is the preferred site of insertion for adeno-associated virus, also known as AAV. And so because of this locus is also known as AAVS1. This particular site is located on chromosome 19, and they showed that insertion of transgenes at this locus has no visible effect on the growth of fitness of many cell types, including primary and immortalized cells, induced pluripotent stem cells, or iPSCs, and embryonic stem cells, or ESCs. And they found that the transgenes, just like with Rosa 26, displayed consistent levels of expression over many cell divisions. So here's an example of that stability from that paper. This group inserted GFP. One of the things they did was they inserted GFP at the AAVS1 site in K562 cells, which is a chronic myelogenous leukemia cell line. And they followed GFP expression over time, which in this case was approximately 24 generations. And even though they did see some variation in GFP expression in both diallelic and pools and monoallelic, the expression was still very stable and didn't go away. And that, again, is not the case with sometimes with lentiviral integration, where you can get silencing due to the fact that you get multiple copies inserted and the locus can be turned off. Okay, so now let's talk a little bit about CRISPR. CRISPR is important because we use CRISPR in these safe harbor kits to insert transgenes. We also use a technology called TALEN, which is an older technology that I'm not going to discuss. But CRISPR, I'm sure you've heard about. It's taken the molecular biology world by storm and is even talked about a lot in the popular press. CRISPR is a very new, although it's been around for a few years now, method for genome editing. The most popular format of CRISPR comes from Staphylococcus aureus bacterium, which is the bacterium that causes strep throat. And it's a two-component system. The first is known as the Cas9 nuclease. And again, this is the most commonly used form, represented by this yellow shape. And the second is a RNA molecule known as a single guide RNA. And this is actually an artificial chimera of two different RNA molecules. One of the RNA molecules provides sequence specificity, as shown by this blue sequence. So this blue sequence is a target site that's supposed to be found, that is found in chromosomes. So basically, if you can program this target site, a 20 nucleotide target site that's found in the genome, into this single guide RNA, this complex will then be guided to that site if it is found upstream of what's known as an NGG or PAM site, protospace or adjacent motif. So again, this is a programmable system. It's also known as an RNA-guided endonuclease. And so when these CRISPR-R gens, as they're known, are guided to these target sites, they cut the chromosome by making a double-strand break. These breaks are then repaired by either one or two mechanisms. The first is known as non-homologous end joining. This occurs when there is no homologous template around to serve as a repair template. And NHEJ is simply the process of sticking the two broken ends of the chromosome back together. Now, this is a highly error-prone process, so what often happens when you get NHEJ, as it's known, is you'll get insertions or deletions, also known as indels, which, if placed into a protein coding region, 2/3 of the time will lead to inactivating frameshift alleles. However, if there's a homologous template around, and this occurs naturally in meiosis or in DNA damage repair, but can also happen when one adds an exogenous molecule of DNA, the cell then uses this homology in order to repair the break. And this is a sample donor clone shown here, where it says donor DNA, which contains sequences that are flanked by homology to the break site. So when these templates are introduced with CRISPR that creates double-strand breaks, whatever information that is placed in between these homologous arms, as they're called, is copied to the donor and therefore knocked in. So researchers have exploited this phenomenon in order to insert virtually any sequence of DNA they want. And this is the type of technology, the type of mechanism that Safe Harbor uses. All right, so let's talk about Safe Harbor, GeneCopoeia's Safe Harbor solutions. I mentioned that we have a number of kits, and again, these are the features of Safe Harbor. This refers to the human AAVS1 and mouse ROSA 26 sites, which ensure transcription competency of the transgenes and presents no known adverse effects on cells. And Safe Harbor integration provides low copy numbers of the transgenes and close to physiological level of expression. So these are our Safe Harbor Knockin Kit components. Basically, we have a number of different kits, but they all have some components in common. The first, and again, I'm only talking about the CRISPR kits right now, although there are tail-end versions. The first component is comprised of plasmid clones that express the CRISPR-Cas9 nuclease and a validated single-guide RNA. And that's true for both human AAVS1 as well as mouse ROSA 26. The second critical component of this kit is knock-in verification PCR primer pairs. I'll show in a little bit how that works. But when one is doing a transgene knock-in using the Safe Harbor system, it's important to do a PCR analysis using what's known as junction PCR at both the 5' and 3' insertion junctions to verify that that the donor construct got integrated into the correct site. Because you can, albeit at a low frequency, you can get random integration. So we do two PCR reactions using primers spanning the homology junctions in order to verify that CRISPR knock-in worked. And the third essential component of the Safe Harbor knock-in kits is a knock-in controlled donor clone. This is a controlled donor clone that contains RFP, or red fluorescent protein. Now, a fourth component, which is optional, although you're going to want some kind of version of this if you purchase a Safe Harbor kit, is a Safe Harbor donor cloning vector, pre-made donor clone, or custom donor clone. So basically, these consist of the plasmid knock-in donor clones that you're containing the information that you're going to knock in to the Safe Harbor site. So I put optional here, but they are an important component that's going to be highly customized. So these might contain an ORF of interest or Cas9 nuclease, or it can be, like I said, a donor cloning vector where you insert a transgene doing do-it-yourself cloning. Okay, so again, I also mentioned earlier that another component of the Safe Harbor system is these Safe Harbor knock-in ORF clones. The Safe Harbor knock-in ORF clones come in this kind of backbone, which contains CMV promoter-driven ORF with an EF1 alpha-driven fluorescent reporter and selectable marker. And this ORF and selectable marker cassette is inserted in between either the AAVS1 or ROSA 26 homology arms. And the features of the Knockin ORF clones collection are that we have over 40,000 sequence verified human and mouse ORFs available. They can be ordered right on our website. And again, these are inserted between the AAVS1 and ROSA 26 sites for ready safe harbor integration. Now, again, some of the applications for safe harbor Knockin ORF clones, I mentioned these earlier, but I'd like to reiterate them. One is rescue of a knockout or mutagenesis phenotype, or overexpression of a fusion tag protein, for example, or expression of a gene from a different species, such as human gene in a mouse. So this data shown here is just to show how well the safe harbor Knockin ORF Knockin system works. This is from the ROSA 26 system. We transfected, mouse neuro2A cells with the safe harbor components and a Knockin donor clone containing both GFP and RFP. And so this shows both the TALEN and CRISPR Knockin kits. When we look after transfection, we see both GFP as well as RFP expression in both cases. We also see that for the control because we selected for puromycin. Control is transfection without the CRISPR TALEN components, just the donor clone. This is expected, but when you do the junction PCR, you will only get PCR reactions if you got insertion at the correct site at the safe harbor loci, at the ROSA 26 locus. And we see that for both the TALEN and CRISPR kits, but not for the control, as expected. We also did some tests on our AAVS1 system. When we do transfection, when we first look at transient transfection, we see lots of green fluorescence, but then when we do drug selection, we see a lot fewer colonies. And this is in the presence of TALEN. And then when we took clones that we isolated from this experiment, we knocked in SOX2 and OCT4. We we used we were able to detect the presence of both SOX2 and OCT4 using gene-specific antibodies. And the control is without knock-in, and SOX2 refers to clones that contained the SOX2 or OCT4 knock-in constructs. You can't see endogenous levels of SOX2 or OCT4 because the expression level, the endogenous expression level in HEC293 cells is too low to be detected. All right, so now I just want to show you how to order safe harbor knock-in components. You can do this right on our website. This is our home page, and in one way you can do this is to go to products and services, and you'll see a drop-down menu which can take you to our safe harbor knock-in kits and clones web page. And if you were to scroll down, you would then see a drop-down box which allows you to order your specific safe harbor knock-in kit of interest. So if you go to species, you would see either human AAVS1 or mouse ROSA 26. So let's say we select human AAVS1. You would then pick CRISPR or TALEN. Again, TALEN is an older technology. Most people use CRISPR, so we'll use CRISPR. And then you have a number of different donor choices that for your custom safe harbor kit. You can order a do-it-yourself cloning vector, a knock-in donor clone constitutively expressing the Cas9 nuclease, or an ORF knock-in clone, or none. You may already have your own safe harbor knock-in donor clone. So let's pick the cloning vector. When you do that, you then get the option to purchase a safe harbor knock-in kit containing a donor cloning vector for human. And you would just select that and add it to the cart. Now, let's say you want to instead just order a safe harbor knock-in ORF clone along with a knock-in kit. You would then go to search by gene up here, And when you see this, you get you see the search tabs for all of our product line, all of our clone product lines. But in this case, you would want to select the tab for ORF Knockin clone. And when you do that, you get the search function for human AAVS1 and mouse ROSA26 ORF Knockin clones. And let's say I want to type in SOX2. So I type in SOX2 and the field begins to auto-populate for both human and mouse SOX2. So I select that and then click search. And then I'm presented with a number of different options. One is for human SOX2 and the other is for mouse. I'm interested in human at the moment, so I'm going to click on the product ID. And when I do that, I get this page, which gives some general genome information about SOX2. But then I want to scroll down, and what I'm presented with is an option to purchase a ready-to-go SOX2 expressing safe harbor knock-in clone. And when I say ready to go, I mean it's ready to use when you receive it. We may have to build it if we don't have it in stock, but that usually only takes a couple of weeks. And you would also purchase along with it the safe harbor knock-in kit, either for Talon or for excuse me, for CRISPR. And actually, I just realized that I selected mouse, not human, but that's okay. The idea is the same. All right, so now I just want to talk about some related product lines. I mentioned earlier Cas9 expressing stable cell lines. This is a related product because some of these cell lines have CRISPR-Cas9 nuclease inserted into either the AAVS1 human safe harbor site or the mouse ROSA26 site. Some do not. Some were built by lentiviral integration. But these are cell lines with stably expressed Cas9. Again, we have more than 40 pre-made cell lines. Most of these are cancer cell lines. Or you can have us integrate Cas9 in your cell line of choice. You can also purchase, as I showed before, the donor clone along with a safe harbor knock-in kit for do-it-yourself stable cell line creation. And these are ideal for several applications such as CRISPR library screening, validation, inducible CRISPR, and more. This is how I mentioned CRISPR library screening before. This is a rough schematic of how CRISPR library screening works. It can be done either in with pooled screening, where cells are infected with pooled CRISPR sgRNA library lentiviral particles, or they can be done individually, where you have individual single guide RNA expressing lentiviruses that are used to infect a Cas9 expressing stable cell line. And this is important, again, because as I mentioned before, CRISPR library screening tends to work best when you start out with a CRISPR stable cell line. And this was illustrated very well in a paper last year where this group, Zlepis et al., transduced cells with Cas9 expressing lentivirus, but they found that the bulk population had some cells that did not express Cas9. And so they ended up subcloning bulk cells in order to get clones with uniformly expressing Cas9 nuclease. So this just reinforces the idea that it's best if you're doing CRISPR library screening to use a Cas9 stable cell line, and you can construct one of these using our safe harbor knock-in kits. All right, so with that, I would just like to summarize. I told you that transgenesis is an important approach in molecular biology with many applications such as cross-species gene expression and mutant gene rescue. I mentioned that some methods for transgene insertion, such as lentiviral-mediated integration, are efficient but can harm cells and are not always stable. I told you about the safe harbor loci in both human and mouse, which provide genomic sites for transgene insertion that permit consistent, stable expression with no known adverse effects on cell fitness or viability. And finally, that GeneCopoeia's safe harbor knock-in kits and knock-in ORF clones for human and mouse provide you with powerful and comprehensive tools for efficient and safe transgene insertion. All right, so with that, I'm going to stop and take questions. Let me just open up the questions box. Okay, so the first question is, Will there be a recording of today's webinar? Yes, there will be. You will get a notice about it in a follow-up email, and you can visit our recordings library. Second, is there a ROSA 26 in human? Actually, yes, there is. It was recently identified, but it is not really caught on yet as a safe harbor site. Okay, third question, safe harbor versus transposon approach in mouse ES cells. Yeah, that's a good question. So you could insert a transgene via transposon. But again, like with lentiviruses, transposons can tend to disrupt genes. So it's kind of risky. Again, safe harbor provides an advantage over it because you're inserting the transgene into a consistent site that's not known to be harmful to cells. What about rat safe harbor? Yes, there is a rat safe harbor, but again, it hasn't been well characterized or widely used. But if you wanted to put genes into the rat ROSA 26 equivalent, you could. Okay, next question. Can I get your PowerPoint? Yes, you can. The slides will be made available for download on the website within a day. Okay, next question. How easy would it be to knock out a gene integrated in AAVS1 site using CRISPR-Cas9? That's actually a good question. It shouldn't be hard at all. The only, a lot of it depends on what the gene that's inserted is. So if it's an endogenous human gene that you inserted AAVS1 and there's already a copy elsewhere in the genome, then you would have to, you might need to make sure that your guide RNAs do not disrupt the endogenous copy. If you only wanted to target the knocked in copy, but it shouldn't be that. Oh, so somebody's saying it's GFP. Okay, then it's no problem at all. Since GFP is not integrated elsewhere in the genome, then there are plenty of validated guide RNAs that target GFP, and so it's not hard at all to do that kind of disruption. Okay, next question. Does your ROSA 26 donor use the native promoter? Actually, it does not. Some people do safe harbor knock-in where they'll have a transgene that's utilizing the endogenous ROSA 26 promoter. That can be true of AAVS1 as well. But the knock-in constructs I showed you, at least the pre-made ones, actually use usually the CMV promoter, or they might use CBH promoter to drive transgene expression. But we can certainly make safe harbor knock-in donor clones that utilize the endogenous promoter for transgene expression. Okay, let's see. Next question. How many bases of homology do you need in the donor? Well, usually the minimum number of bases on each side is about 500. People tend to use longer lengths of homology, like maybe a KB or so. But it's usually anywhere from 500 to 1,000. Okay, next question is, do you have safe harbor gene knock-in kits for other species like bovine or sheep? That's a good question. Not at the moment, but we are always looking to add new products. And again, if you wanted to order such safe harbor reagents from us, we can definitely build them for you. Okay, so are there any other questions from anybody? Anybody? All right, well, if not, then I would like again to thank you all for joining me for today's webinar. If you have any questions, go ahead and give me a call at this number, 866-360-9531, extension 227, or feel free to send me an email at edavis@genecopoeia.com, and go ahead and visit us on the web, www.genecopoeia.com, where you can check out all of our CRISPR-related products and services, and as well as our other non-genome editing products and services. So just check us out and see what we've got. Anyway, with that, I'd like to again to thank you very much and to have a great day.